Hello friends, welcome back to my channel Biofric. In today's lecture video, we will be talking about Calvin cycle or C3 cycle or reductive pentose phosphate pathway, or another term here is the dark reaction, or also it is sometimes referred to as Calvin-Benson cycle. This Melvin Calvin and Benson, these two were the scientists who basically elucidated this whole process, and they got Nobel Prize for that. So we are focusing now on the dark reaction. We saw the light reaction in the previous classes. Light reaction gave us two major products, that is, one is ATP and the other one is NADPH, right? So both these products will be utilized now in the dark reaction for the carbon fixation. How that is done? So let's look into that. So to start with, we need to know where does this dark reaction occur. This occurs in the stroma. I mentioned this even before, right? So in the stroma, your dark reaction occurs. And overall, your dark reaction has three different phases. Okay, so there are three different phases. First being carboxylation. Second is reduction. And the third one is regeneration. So these are the three major phases, and let's look at them one by one. So let's first begin with the first phase here, that is carboxylation. As per the name, we are adding carbon dioxide in this whole phase. Okay, now we start our dark reaction by taking three molecules of RUBP. Now, what is this RUBP? RUBP is nothing else but it is ribulose 1,5 bisphosphate. Okay, now this ribulose is a 5-carbon. RUBP is a 5-carbon compound here. Now, in this process, the first step that occurs is one proton moves out. One proton moves out. And here comes our enzyme Rubisco. I'm sure you guys have heard this name, Rubisco. This enzyme is one of the stupidest enzyme. Again, why am I saying stupid enzyme? Because of its the way it acts. Okay, the Rubisco enzyme here is also known as ribulose 1,5 bisphosphate carboxylase and oxygenase. So it is very much confused from its name itself. You can say that it sometimes shows you the carboxylase activity, sometimes shows you the oxygenase activity. So for that matter, I'm saying it's one of the stupidest enzyme, but being stupid, it can no other enzyme can do its function, and it's very important as well. And in addition, it is one of the most abundant enzyme. In fact, it's the most abundant enzyme in the chloroplast or on the planet Earth itself. Okay, now this Rubisco is acting upon RUBP, and the first intermediate that we get here is inidiolate. So this intermediate is unstable intermediate. Most of the intermediates are quite unstable. So this inidiolate that you have here gets converted to something called as 2-carboxy 3-keto d-arabinitol 1,5 bisphosphate. Okay, bisphosphate. So now how do we get this compound? There has to be some addition of some sort of compound there, some sort of molecule there. So basically we are adding carbon dioxide here. So that's the name, carboxylation. So we add the plant takes in the carbon dioxide, and in this reaction carbon dioxide is added. So 2-carboxy 3-keto, so there is a keto group there, d-arabinitol 1,5 bisphosphate is the product here. So from here, in the carboxylation phase, the last end product that we get here is something called as 3-phosphoglycerate. Okay, so what do we get here is 3-phosphoglycerate. Now, how many phosphoglycerates do we get here? Since we are using 3 molecules of RUBP in the start, okay, which are 5 carbon each, and then we are adding carbon dioxides, which are again 3 molecules of carbon dioxide should be used. So 1 RuBP uses 1 carbon dioxide molecule. So for 3 RuBPs, we need 3 carbon dioxide. Overall, in this whole chain, we have 18 carbons. So from these 18 carbons, we can, somehow rearrange them, and this 3-phosphoglycerate is a 3-carbon compound, and we get 6 molecules of 3-phosphoglycerate in the carboxylation reaction. So I hope this carboxylation steps are clear. This is it. So to brief upon this again, carboxylation is we are basically adding the carbon dioxide here so that we take in those extra carbons. See, ultimately you will see that in the regeneration phase, that's the third step, we'll be regenerating the RuBP. But then you will get one sugar molecule as 3 carbon sugar, okay, because we are going for the 3 molecules of RuBP, right? So those 3 molecules of sugar, the 3 carbon sugar will be later utilized, and that will be used in the production of sucrose or starch. And other remaining molecules, that are 5 molecules that you will get, those will be regenerated again to form your RuBP. This you will understand slowly, even if you are not understanding this at this point. Just remember that we are starting with the 3 molecules of RuBP. Rubisco is the enzyme that is showing what the activities here. It will show the carboxylase activity. Then inidiolate will be converted to 2-carboxy-3-keto-d-arabinitol 1,5 bisphosphate by addition of that 3 molecules of carbon dioxide. And from there, you basically add water here to get your 3-phosphoglycerate. 3-phosphoglycerate, such that we will get 6 molecules of 3-phosphoglycerate. Now, I would like to show you something about the Rubisco enzyme as well in the side. Okay, so let's begin with the let's talk something about the Rubisco enzyme, why it's so special, and what is there in it. So this Rubisco enzyme that I'm talking about is ribulose 1,5 bisphosphate carboxylase oxygenase, right? So it's a pacemaker enzyme, and it has overall 8 large subunits. Okay, so it has 8 large subunits and 8 small subunits. Large subunits are coded large subunits, these are coded by the your chloroplast genome. So it's coded by the chloroplast genome here. And this smaller subunit, smaller subunits which are there, these are coded by the nuclear genome. Okay, these are coded by the nuclear genome. Okay, so now the Rubisco enzyme depends on the concentration of carbon dioxide, oxygen, okay, because we know that it shows both the activities, carboxylase and your oxygenase activity, right? So besides that, it also depends on the magnesium ion concentration. Why magnesium ion concentration? I'll tell you, because it is important to basically activate your enzyme here. And besides that, the optimum pH is required for its activity, that is 8. Okay, so pH 8 is expected to be there so that you activate the enzyme. Okay, now let's talk about making this Rubisco enzyme active, because it's not always in the active form. So now what I'm saying is that you have your enzyme Rubisco, something like this here, and this enzyme Rubisco has a lysine residue on 2-not-1 position, and this lysine residue has NH3 plus group. Now it is in the protonated form, right? So this will be deprotonated. So basically that one proton will be taken out, and you will have your enzyme Rubisco, your lysine, and this will be deprotonated. Now you might wonder, is it active now? No, it's not yet active. So what else is done here? Now we add carbon dioxide. So basically this enzyme also requires, interestingly, carbon dioxide to be added. So we add another carbon dioxide, and another protein is released. So we can see that there are another proton is released out of that, and we have NH2. So one proton from there is released. As a result, what do we get is Rubisco on this enzyme Rubisco. We have lysine and we have NH, and since we are adding carbon dioxide, it will be COO minus. Now this is called as carbamate. This is called as carbamate. Now this carbamate is formed basically by this whole step here that is called as carbamylation. Okay, this is called as carbamylation. Now you might ask me, is it active now? No, it's not yet active. So it requires another factor here, that is magnesium, that's added. So now once you add magnesium to this, it becomes active, but at the same time it also requires light for its activity. So you might ask me, like, this is dark reaction, why do we need light here? But then my dear friends, it's not really the dark reaction taking place in the dark, but basically the light reaction is taking in the light, and it's absorbing the light. It's exactly dependent on the light, but this reaction is dependent indirectly on the light. And here if you go to see, you actually need some amount of light as well. Okay, now why am I saying this? Again, I'll clarify this further. So now your Rubisco enzyme that you have, right, this is one way of activating your enzyme. You require light. There are certain nocturnal inhibitors that are sitting on your Rubisco enzyme here. Okay, so you have your Rubisco enzyme and this nocturnal inhibitors, like there is one nocturnal inhibitor, carboxyarabinitol 1-phosphate. Now this carboxyarabinitol 1-phosphate is a nocturnal inhibitor. Okay, and it basically binds to this Rubisco, inactivates the Rubisco in the dark. Once it senses the light, it is broken down and your Rubisco gets activated, or it can be also again activated by the enzyme Rubisco activase. So there is another enzyme that is important here, that is Rubisco activase. Now there is another function of this Rubisco activase besides doing this job. Okay, see, your nocturnal inhibitor can be removed either by the light or your Rubisco activase, and Rubisco activate itself has this one function of removing the nocturnal inhibitor if it has to be removed. And importantly, when you have your enzyme Rubisco that is bind to that is already bound to the RUBP. This is your enzyme and this is your substrate, that is RUBP. See, Rubisco is the enzyme that is required for this whole reaction, right? So now if your substrate binds to the enzyme, okay, that is not active. This enzyme that I'm talking about is not yet active. It is for its activation, carbamylation was important, isn't it? But now this enzyme is not yet active because a carbamylation is not occurring and it is already attached to the substrate. So it won't get active because this is blocking, the substrate here is blocking the site for carbamylation. So here comes your Rubisco activase. And this Rubisco activase will help you. Rubisco activase, for that matter, I'll write it in short. So this Rubisco activase will utilize ATP. Okay, it will utilize ATP and it will free your enzyme from RUBP. Okay, so RUBP will be kept aside and enzyme will be free. Now it will be carbamylated. Okay, again the magnesium ion will be added. Now later it can bind to the RUBP. Here again it requires ATP. This enzyme, it requires ATP and light. So in short, the concluding factor is dark reaction is not really taking place in the dark. Okay, so I hope this was about the enzyme here, and we actually spoke about another factor here, that is Rubisco activase, then again the nocturnal inhibitor concept. So I hope this extra concept that we saw here is also clear, because it's important while learning the carboxylation itself. Okay, because this Rubisco enzyme plays an important role only in the carboxylation. Now let's move ahead. Let's talk about the reduction phase. Okay, now in the reduction phase, which is the second phase, what do you think will be happening? In the reduction phase, we are actually using those ATPs and NADPH. Okay, now in the reduction, you remember what was the product here? 3-phosphoglycerate, and like that we had 6 molecules. So I'll write 6 into 3-phosphoglycerate, right? So this 6 molecules of 3-phosphoglycerate will be now converted to, or basically it will be phosphorylated to 1,3 bisphosphoglycerate. How this is done? Can anyone think and or write it there without me telling. Just try if you guys want. See, it's you're adding phosphate. So as I said, we will also use ATP here. So basically, we are using all 6 ATPs. Okay, since we have 6 molecules, we'll require 6 ATPs, and you will get 6 ATP formed here. And which enzyme is helping in this is 3-phosphoglycerate kinase. So we are adding phosphate on this. So 3-phosphoglycerate kinase, right? So we're adding phosphate. Now after adding phosphate, we will be basically doing the reduction. Okay, how that is done is we'll use our 6 NADPHs here to form 6 NADP plus, and those hydrogens will be used to reduce the compound here, that is 1,3 bisphosphoglycerate to glyceraldehyde 3-phosphate. So now here we will be getting 6 molecules of glyceraldehyde 3-phosphate. So this is the end product of your regeneration phase. To summarize again, in the carboxylation phase, we just added carbon dioxide and we obtained 3-phosphoglycerate, 6 molecules of 3-phosphoglycerate from 3 molecules of RUBP. Here in the reduction phase, what did we do was we utilized your ATPs and NADPs, okay, to reduce your compounds here to obtain 6 molecules of glyceraldehyde 3-phosphate. Now comes your third phase, that is regeneration phase. Let's go to that. Now this is a bit tricky and it might be confusing, but then pay attention from the beginning. So now what is the end product here? 6 molecules of glyceraldehyde 3-phosphate. Now let's get started. Okay, now take a fresh page and I would recommend you to write it down. So glyceraldehyde 3-phosphate for sure. I have 6 molecules. Out of the 6 molecules, 1 molecule will be used in the sucrose or sugar formation. Okay, so basically this is 3-carbon compound, right? This is 3-carbon compound that I have, glyceraldehyde 3-phosphate. That will be used in sucrose. 1 will be used in sucrose. 5 will be used to regenerate what? RUBP. How will I do that? So let's first start with the 5 molecules of glyceraldehyde 3-phosphate. Okay, now I have 5 molecules of glyceraldehyde 3-phosphate. Out of this, I'll use 2 molecules of glyceraldehyde 3-phosphate. Now here, what is the balance? Here I have still 3 molecules of glyceraldehyde 3-phosphate if I use 2 molecules. So here, first and second, I am utilizing glyceraldehyde 3-phosphate to get something called as DAP. What is this DHAP? This is dihydroxyacetone phosphate. Okay, this is also one of the intermediates in the glycolysis cycle, if you remember. So now this DAP that I have, okay, the 2 molecules of DAP I have formed. Now what do I do is that I obtain another molecule of glyceraldehyde 3-phosphate. This is the third time I'm using this. So I'll take glyceraldehyde 3-phosphate here. And now what do I do is that I'll combine both these and obtain something called as fructose 1,6 bisphosphate. Okay, you guys can write bisphosphate. I'll write it for short because it's a long chain there that will be formed. So DAP and glyceraldehyde 3-phosphate are combining here to get fructose 1,6 bisphosphate. Now you can remove one phosphate group, okay, and you will get something called as fructose 6-phosphate. Now once you get your fructose 6-phosphate, what I'll do is that I'll use another glyceraldehyde 3-phosphate. Okay, so I'm taking another glyceraldehyde 3-phosphate here. This is now the 4th time I'm using, right? So I have just 1 glyceraldehyde left with me. Now I'm using glyceraldehyde 3-phosphate, which is a 3-carbon compound. Fructose is how many carbon? Fructose is 6-carbon compound, isn't it? So the fructose is 6 carbon. I have 3 carbon, 3 and 6 total. I have 9 carbons here. What do I get from this? From glyceraldehyde 3-phosphate and fructose 6-phosphate is that I'll get 2 compounds after rearrangement. One is erythrose 4 phosphate, and another one is xylulose 5-phosphate. This is a 4 carbon, so 4 phosphate, just remember it like that. It's not necessarily 4 phosphate, that means it's 4 carbon, but it's easy here to remember. Xylulose 5-phosphate is 5 carbon compound here. Now what happens is you have erythrose 4-phosphate, which is 4 carbon, and xylulose 5-phosphate. Now we'll be following 2 chains here. Now this DAP molecule that I had here, another DAP molecule which I had, will come here. I'll write DAP here. This DAP and erythrose 4-phosphate that I have here, what will they do is that this is 3 carbon, by the way. DAP is a 3 carbon. This is 4 carbon. 4 and 3, I'll get a 7 carbon compound here. That is pseudohaptulose 1,7 bisphosphate. From here, I'll remove one phosphate group. I'll get something called as pseudohaptulose 7-phosphate. Now, once I have this pseudohaptulose 7-phosphate here, I'll take another remaining glyceraldehyde 3-phosphate. Okay, I had 5. I used 4. Okay, 1 is still remaining. I'll take that here along with pseudohaptulose 7-phosphate. So I'll write glyceraldehyde 3-phosphate here, and now what I'll get is again 2 different things. So again now I'll get something called as xylulose 5-phosphate and ribose 5-phosphate. Both are 5. So 7 carbon was pseudohaptulose 7-phosphate and glyceraldehyde was 3 carbon, total 10 carbon. If I rearrange them, both can be 5-5. Okay, so I'll write ribose 5-phosphate, and another compound that I'll get is xylulose 5-phosphate. From this ribose 5-phosphate and xylulose 5-phosphate, I'll obtain something called as ribulose 5-phosphate and ribulose 5-phosphate. Okay, here it is epimerase enzyme, while here it is isomer. So isomerase enzyme will be used. So from this ribulose 5-phosphate, what I can derive is ribulose 1,5 bisphosphate by using one ATP here and using another ATP here. I'll get another RUBP formed. One more is still remaining. Still we have to use another ATP now, and one more RUBP has to be regenerated. We have regenerated just 2. So here you remember xylulose was still remaining. So from that xylulose, what I'll form, I'll be following this similar path again. So from here, using epimerase, I can form ribulose 5-phosphate. I'll use ATP to ADP here, and what I'll form is RUBP, that is ribulose 1,5 bisphosphate, isn't it? Now I want you guys to remember certain important points. This looks very messy. You guys can draw this on your own step by step, and you will understand how this whole regeneration occurred. Basically, we have regenerated all these 3 RUBPs which we had utilized in the first step here. Okay, now how many carbon dioxide did we use? 3 carbon dioxides were used here for 3 RUBPs. Then we utilized 6 NADPHs. We utilized 6 NADPH. Then we utilized how many ATPs? 6 ATPs here, and plus these 3 ATPs that were used to regenerate. So total 9 ATPs were used. 3 carbon dioxide molecules were used, and we regenerated all 3 RUBPs. And net we got just 1 glyceraldehyde 3-phosphate that was later used for the sugar formation or starch formation. So basically this is how all the dark reaction occurs, and besides that we also learn the enzyme Rubisco and its activation. So that was it, guys. I hope this was helpful, and see you in the next lecture.